This is a production of Cornell University.
Yeah, thank you for the detailed introduction. Amazing. And first of all, I need to say that thank you for attending. It's great to have you as audiences, and I'm excited to present my work. So my work is about glyphosate, and I'm looking at glyphosate interaction with organo-mineral association, and focusing on kinetics and mechanisms. This is what we know about herbicide application in the United States. Glyphosate is most heavily applied between all those herbicides to kill weeds. And over the years, it's became more popular, and farmers love this herbicide. And geologically, if you look at this map here, so this is the Mississippi River Basin. It's Alps. And Central Valley, Columbia River, and Washington State. Those are plains which heavily farmed and heavily used for herbicide. Herbicide, or you can know as this trademark, Roundup, it's became a little bit controversial in terms of environmental toxicity and occurrence. We can find everywhere in soil, water, even air, and also it can be accumulated in plants. However, it's not carcinogenic. It's defined by EPA. In terms of like a usage, that's a thing. We are thinking we should more pay attention about that. I'm showing here the most the main processes of glyphosate in environmental compartments, how it moves around. After spray, it can be by accumulating plants, or it can be degraded by UV lights, or it can enter into soil for water and interact with minerals, specifically organo-mineral associations. Excuse me. I'm interested to learn about how it retains and be released or transforms on those mineral and organo-mineral association abiotically. And also, it's really important to know how these byproducts can be generated after transformation, because we see AMPA, ammonium-methylphosphoric acid, which is one of those byproducts. This can be uptaken by plant much more than glyphosate. And also it has the same toxicity impact on cells. This is a general understanding and general figure of my dissertation. Organo-mineral association concentration diminishes as we go down the profile. And after spray, obviously it's going to interact with organo-mineral association on top. In terms of interaction and type of organo-mineral association, it can go into the retention process, or it can be transformed. So manganese oxide is very well known for transformation or oxidation of glyphosate. So this is a candidate for doing that study. And iron oxide is selectively absorb organics and form organo-mineral association. So it's a good representative to pick as an organo-mineral association to see glyphosate retention. Act 1. In Act 1 or Section 1, it's a kind of into section for my dissertation. And that's about wettability of particles in soil. Organo-mineral association forms, and obviously can change the wettability of that. In the other language, we call them how it can be more hydrophobic or hydrophilic. If we can measure it, is measuring water contact angle. If the contact angle is about 90 degrees, it's going to be more hydrophobic, and below that is hydrophilic surface. So goethite is a hydrophilic surface, and I picked diamond because we have diamond plate, and that's a hydrophobic surface. It can be used as a model to study how organomineral association formation can change this hydrophobicity or the philicity on both surfaces. So for organic or dissolved organic matter, I picked polysaccharide. Polysaccharide can be found in soil, and it comes from plant cell wall and also microbial necromass. And they accumulate at the surface of the minerals, specifically on metal oxides. And what is interesting, so now I have a model system. I can see that more fundamentally instead of using like the extraction of DOM. On this graph here, first of all, I use in situ ATR spectroscopy is a technique to see absorption at the surface of a mineral. If you have a mineral, you can introduce your organic, your target sorbent. They're going to absorb, and you're going to see some peaks. They come different frequency, and those frequency is related to different bondings in each molecule. At the surface of the goethite, we see absorption of polysaccharide is much more than diamond. That's because polysaccharide itself is hydrophilic, and hydrophilic loves hydrophilic. In terms of kinetic, with increasing surface loading for polysaccharide, we notice it gets slower, and we have more adsorption. And subsequently, when we have desorption phase, some of those adsorbed can be desorbed. If it's much, we have more adsorption, there's more chance for desorption. But in lower adsorption rate, we have less desorption. The other way to translate that, looking at isotherm, adsorption, desorption isotherm also shows this loading at different scale, like a phase, adsorption and so desorption. This is a similar data set for diamond. So we have less adsorption compared to goethite, kinetically much faster. And during desorption, I can say most of them are desorbed, and some of them are still at surface. And this is, you can see an isotherm as well. This is really important to know first how much stuff you have, how does it look like the loading, rate, and capacity. What I did next, I was interested to see the hydrophobicity and philicity. I used water contact angle measurement meter to measure water contact angle. With increasing surface loading here, we notice on both surface, hydrophobicity increases since we have higher contact angle. That was interesting. But it doesn't matter what surface you start, but you load more, surface became more hydrophobic. And this picture here, it's not obvious actually, maybe that screen is much better. The outline here, this space is loaded by polysaccharide. This is a TA image of polysaccharide goethite association. That's giving you a little bit visual result. How does it look like? In summary, and also more data we have here in terms of functional interaction of functionality on polysaccharide with surface. What we learned in the interfacial sequence for those functional groups, you can see functional groups here. This is polysaccharide structure, carboxyl groups, amide groups, and metal ester groups, and this is backbone. If it's the surface is diamond, a hydrophobic surface, backbone, which is a more hydrophobic surface, hydrophobic component, will start to show up at the surface. But loading more and more, we started to see amide and carboxylic functionalities. Those are more hydrophilic. But at the end, it's wrapped up by a metal ester functional group, which is hydrophobic, more hydrophobic than other functionalities. We have the same trend, like in terms of how this hydrophilic functionalities accumulate at surface. On goethite, at the end of with loading, ultimate loading, beside getting into luster. And this surface again became hydrophobic on the top here at most surfaces. This is very important to know how much organic matter can change the hydrophobicity or hydrophilicity, and how this gonna impact the transfer of contaminants when they're gonna interact. Act 2. In Act 2, so I introduced glyphosate into the system, and I found, that was the question actually. The question was how this organic complexation now can change glyphosate retention, or if there's any potential for release when they absorb or not. This is a batch study data I'm showing here. The first, look at this plot at the left side. I think this red line showing absorption during, showing absorption of glyphosate with increasing loading. We found with loading more polysaccharide, absorb less, glyphosate absorb less. And after, when we had desorption, this is the blue line, we had some desorb, but the same trend, If you look after a specific point with loading more polysaccharide here and here for absorption and desorption, we had a little bit increase for glyphosate absorption. It's more favorable, it looks, to have more glyphosate, even we have a lot of polysaccharide at the surface. What I did, I said maybe it's about like a charge. We can look at the charge of the surface because electrostatically we are interacting each other. I measured the potential of those samples. I found with increasing this this green line here, with increasing loading, we are generating more negative charge. Glyphosate itself is zero ion, but in total is one is a -1. So that means we should have less absorption, and that's which is about this part. So what about this part? When we have more loading. When we have more loading, it looks, when we add glyphosate in, this is the red dots, the zeta potential became a little bit more positive, still negative, but less in terms of repulsive force, it's less repulsive. And we can have more interaction, attractive interaction. Fundamentally, I have one more question. What is happening? Like, why do we see this increase for charge? So I looked a little bit in detail using STD-NMR technique. STD-NMR technique is a very good technique to study host-guest complexation between protein and small ligands. This is the combination of glyphosate and polysaccharide. We had a sample. This is just glyphosate only. We found when we have peak of glyphosate in this sample, when we have a STD-NMR spectra, that actually shows we have non-covalent interaction via hydrogen bonding between polysaccharides and glyphosate. They can interact with each other through hydrogen bonding. And hydrogen bonding is a very good sort of interaction to stabilize the complex. So the complex can immobilize easily at the surface. So we have this increase now. Oh, sorry. That was a batch study. Now I want to see more about glyphosate itself and its bonding, how the molecule interact at the surface, and how those functionalities in glyphosate. We have glyphosate here. It has phosphonate group, amine group, and carboxylic acid group. I want to see how it is interacting at the surface of polysaccharide goiter association and also goiter. So at the right-hand side, we are looking at PO band, which is related to this interaction, absorption of glyphosate. And at the right-hand side, you have carboxylamine couple, this peak that shows this part, the green part of the glyphosate. And I looked at peak area versus time. This is the thing you can figure out how much stuff you have and how it changes as a function of time. So this is for PO band, and this is for carboxylamine couple band. So third thing we can find, so this is faster. Carboxylamine functionality is interacting faster, but this phosphonate is not interacting at the same rate. Another thing, with increasing PS loading this way, I'm loading more polysaccharide, we decrease both functionalities, and that's equivalent to less absorption. The reason to have two different rates for one molecule is actually about interfacial species. We don't have only one type of glyphosate at the surface. We have multiple different types, and those are in different locations. And in total, in general, we see different rates. Oh, one thing I should mention that. Okay, and if you look the type of binding, this is bidentate binding. We have two oxygen and connected to two iron atom. And here only we have one hand, which is or one bonding. We call them monodentate. So when we have polysaccharide, monodentate bonding is more favorable. And that's really important because bidentate is much stronger than monodentate. And that means there's a chance that glyphosate can be released from polysaccharide-goethite association versus goethite, bare goethite. Again, I like to dig more in detail, and I look at the carboxylamine versus carboxylic acid versus amine group. So I deconstructed the peaks, and this is the deconvolution analysis. And I found if I look the the contribution of this functionalities, I can find how much they are important in interaction during adsorption and desorption. What I found during adsorption, with increasing polysaccharide loading, ammonium, this functionality here, this functional group, have getting more contribution in the interaction with the retention of glyphosate. And that's really interesting for me. And it makes sense, actually. Actually, amine is positively charged. So electrostatically, they can interact more with the negative charge components in polysaccharide. And it makes a tertiary complex, polysaccharide, glyphosate, iron oxide. And we have a chemisorption from the one side, and we have a hydrogen bonding from the other side. So this way, glyphosate can be stabilized at the surface, and then maybe it can have delayed release when maybe have more precipitation in off-season, during fall or winter. In summary, I know now you know why glyphosate dances on genital floor. It's more mobile. Looks. So glyphosate retained to less extent, we can find from just counting, so just comparing those peaks. And polysaccharide-goethite association, that means we're going to have greater transport of glyphosate into water. And polysaccharide association promotes more monodentate binding, which is less strong. And the retained glyphosate, that's the kind of interpretation from those data that says that could be possible to have a delayed leaching. Act 3. In Act 3, I have a different question. In this question, I'm looking at how low molecular weight this time. Polysaccharide is high molecular weight compound in soil. But I wanted to know how low molecular weight dissolved organic matter can interact with glyphosate and how it changes their absorption and retention. So I made a binary system this time. I mixed them together and let them to absorb. So for low molecular weight DOM, I'm using this peptide. It has 19 amino acids as a model peptide. So before starting to study absorption on GO-type, I was interested to know, is there any interaction in solution itself, this low molecular weight and glyphosate can interact? So I used two different techniques, IR and NMR. So in IR, and at the left-hand side, this spectrum showing glyphosate plus peptide sample. If you compare the lines, the dashed lines, which is from glyphosate peptide, you can see this is the summation of those spectrum with no shifts for frequencies and no shifts actually for broadening or shortening of the bands. And that means there's no interaction. We tested again with a different technique, and we looked at those amino acids this time. This is the NMR toxicity for peptide sample, which is those black region, and peptide plus glyphosate sample are showed by red regions. If you look those regions from peptide and peptide plus glyphosate, they're overlaid, completely covering each other, and there's no differences. They are not moving around. That means there's no interaction. What about surface? What about the surface? I looked at glyphosate peptide absorption spectra and to know how peptide can change the absorption rate and capacity, and also how it changes its complexation. The data shows here, I looked at this bonding here, which is a POFE bonding for chemisorption of glyphosate. Glyphosate itself is the signature, this is the curve for glyphosate itself. Plus peptide, you can see dramatically dropped. We have less absorption for peptide. They are competing for surface, and peptide is winning this game. Peptide can sit, occupy the surface, and didn't allow glyphosate to be absorbed. Another thing is about kinetics. Also, kinetically, glyphosate absorption is slower when it's in a solo system. But when we have peptide, it's became faster. The reason is chemisorption kinetically is a slow reaction, but outer surface complexation without chemisorption is faster. So that means peptide is changing binding type, not only absorption capacity. Also, we found if there is any type of chemisorption for glyphosate with peptide, it's more monodentate form, which is a less strong one. In the summary for that, I can say peptide, glyphosate absorption in the presence of peptide decreases the absorption for glyphosate. And that means we're going to have more transport. When we have low molecular weight, kind of soup, and plus glyphosate are not able to sit and find some place for it, sit for itself. So it goes away and stay in water. And other things, as I mentioned, low molecular weight DOMs like peptide promotes more monodentate and outer sphere complexation. Those type of binding are very weak bonding and also less strong. And in general, we found there is no interaction in solution and at the surface between glyphosate and peptide. That was another, which is opposite to polysaccharides. High molecular weight, they interact with each other. Then you have glyphosate high molecular weight compound, but low molecular weight and glyphosate, they do not interact. The last act is about transformation of glyphosate. So I looked at adsorption and desorption. Now the question is how organ-mineral association impact transformation of glyphosate. Glyphosate transformation, I think I mentioned this, create amino-methylphosphoric acid, which is toxic, as same as glyphosate, for early stage kind of cell development in animals and plants. And so we should study amino-methylphosphoric acid more, and we look at that. So and this compound originates from transformation. So I wanted to know how organ-mineral association can change generation of this product or transformation of glyphosate. So the model mineral is manganese oxide. We have two manganese oxide in this system. Behrooz and osmionite. Your manganese oxide, the differences is they're having different catalytic capability. The reason is having different content of manganese 3, 4, or different manganese, different oxidation states for manganese. Behrooz have more manganese 3 and 4, but osmionite has more manganese 2 and 3. That means lower average oxidation state, lower catalytic capability, or oxidative transformation for glyphosate. On this slide, I'm showing two interesting pathways in terms of my study. The pathway that create amino-methylphosphoric acid, or AMPA, and glycine. Glycine pathway, and the reason we have two different molecules because this cleavage, a CN cleavage, can be happen in two different positions, and they can create two different molecules and two different pathways. Either is it AMPA or glycine. Two steps are really important steps for transformation. The first step is surface complexation. Glyphosate absorbs. And then we have a single electron transfer reaction. Electron jumps into the surface. And then we have a radical species. And this radical species start to degrade. Glycine is okay. If you have glycine, only this pathway, we should be fine. Glycine is safe. It's amino acid. But the AMPA is not safe. And also it accumulates more in plants. So this pathway is a bad pathway, and this pathway is a good pathway. Again, is there any interaction between protein and glyphosate before starting to see the interaction at the surface? Protein is high molecular weight dissolved organic matter in soil. So I was expecting to see interaction, and I saw it. STD-NMR showed this interaction. And this time, the interaction was much stronger. But now covalent bonding between glyphosate and protein, shows better signal for STD-NMR. The bigger signal means stronger non-covalent bonding. So let's talk about adsorption first, and then we're going to talk about transformation. So the first step I said is surface complexation. For this, I use in situ ATR, FTIR spectroscopy. Since I have a very limited surface on ATR, I'm able to only detect glyphosate adsorption. We have a lot of glyphosate and limited surface. The signal from byproduct molecule is negligible, and it's not overlapping or interfering with the glyphosate signal. So I can draw some data based on what I'm seeing and referring that to POMN bonding. This bonding here is a bidentate bonding between manganese oxide and glyphosate. On top, we have Behrooz, this panel, and this panel osmionite. So I'm going to talk about Behrooz first. First two lines from top are showing the experiment at pH 4.6 without and with BSA protein. And the next two, which are lower, showing lower absorption, they are pH 7.2. So this is the first result. Increasing pH, decrease absorption. And that makes sense. Glyphosate became more negatively charged, and surface also gets more negatively charged because those manganese oxide having lower ZPC, 0 point of charge, and when the pH is above that, surface became more negative, and we have more repulsive force. What about BSA? BSA added in and to the system decrease absorption. We have less absorption now when we have BSA at surface. It doesn't matter what pH is that, or even the surface. We see the same trend even for husbandite. And husbandite also showed this less absorption capacity when we add BSA in. And also the same trend for pH. So BSA association reduced the extent and absorption rate for glyphosate on both coated manganese oxides. At higher pH, I said less capacity, absorbing capacity. That means, so we're gonna have more into the water. For transformation, I needed to extend the surface. So I wanted to measure byproducts. My surface needed to be bigger. So I designed a new system, a new kind of experimental design. So actually I'm mimicking the transport in pore water. These are soil particles, and also soil particles coated with manganese oxide or iron oxide plus organic matter. So this is what I did. I made BSA manganese oxide chips. Protein is loaded onto the manganese oxide here, and this glass slag goes into the microfluidic reactor. Then we introduce glyphosate in. As a function of time, we collect effluents and we analyze for byproducts using LCMS or other photometric approach. This is a closer view to the burner site and husbandite chips. So here on top, this is a cross section of that. It shows how thick is film, the manganese oxide, and for husbandite, for burner site is around 2 micrometer, and for husbandite is 4.5 micrometer. So they have different amount of manganese oxide at the surface, but the thing is very important is the surface area. Internal surface area or total catalyst surface area, we have almost similar surface area now. So they're gonna have a fair condition in terms of surface area. And these two are TDM images of those crystal kind of particles. Now we are looking at breakthrough curves from those microfluidic reactors. When we have burner site, and then in the next slide, we're going to look at husbandite. So I sort the result based on pH here. At the left-hand side, we have pH 4.6. At the right-hand side, we have pH 7.2. On the bottom, on bottom of the panel, we have burner site with BSA, and on top, burner site alone, no BSA. It's very obvious, like with increasing, at greater pH, transformation became less. You have less byproducts. And also, if you compare at either like pH 4.6 or pH 7.2, when we have glyphosate, when we have protein at the surface, so it diminishes the degradation. We have less transformation for glyphosate. In terms of pathways, I calculated how much of transformation related to AMPA and how much is related to glycine. I measured, since I have data for AMPA and glycine. At pH 4.6, it looks they are going equally. We have both pathways, and they are almost equal, correct. Increasing pH, it changes the game. Now, glycine pathway gets more dominated. Now, is primarily, we can say it goes this way, around 70% and 30% that way. We have two numbers here because of the with BSA and without BSA. With BSA protein at the surface, we see more AMPA as a pathway. As a little bit change. If it's 40 without BSA, it became 46. Here at pH 7.2, if it was 28, now it became 32. So BSA association kind of changed the pathway toward AMPA, but it's still glycine more. Exactly same trend, not exactly same, similar trend observed at the surface of azimlite with and without BSA at both pH. And here, but the ratio between AMPA pathway and glycine pathway is not the same. It looks azimlite mediate more glycine pathway, 80% at 4.6 and 86% at 7.2. And there's no difference with having or not having BSA. They have exactly the same ratio. And one thing I didn't mention that is about if there's any chance to go further transformation for byproducts, the way we can say that measuring ammonium, and I didn't detect ammonium at the surface of azimlite, but Brunner size shows a couple percent, like a 2 to 5% ammonium. That means still Brunner size more catalytically active to degrade glyphosate one more step to get ammonium, phosphate, carbon dioxide, and water. So here, I'm summarizing the protein association impact. So it looks active site occupied by Brunner site, by BSA protein. Then what's happening? So it limits the absorption and subsequently limits transformation rates. Another thing that we found from STD-NMR study, there is organic-organic interaction. So how it changes the system? If you have organic-organic interaction, so glyphosate has less chance to interact with the surface and to be degraded. So it protects glyphosate from fire. So another thing that's interesting, this is studied by many people, and they found protein can immobilize and reduce manganese. When we have reduced surface, and that's another way to have less transformation, because catalytic capability drops when we have reduced surface instead of having oxidized surface. Here, I have time. This is the thing I'm proposing for surface passivation of manganese oxide. So I measured pH and also dissolved manganese, soluble manganese in effluents to learn how it changes. Because when we have glyphosate transformation, we have an electron transfer from glyphosate to surface, and surface became reduced. That means manganese oxide became transformed. So we have not the same surface. The surface changes when we have more transformation. And the transformation causes higher pH. That means we're going to have high pH in solution that diminishes the transformation. We have more soluble manganese in solution. That means reduced surface. Again, it decreases transformation. Another thing, we have glyphosate. Glyphosate can complex with manganese, reduce manganese, and make a dissociation for surface that we call ligand promoted dissociation. And ligand promoted dissociation decrease the catalytic capability for manganese oxide. And another way to passivate surface. So this is a negative feedback loop. In long term, if you run this experiment, degradation of glyphosate is going to become less, and less. So I'm going to summarize entire 4 acts. So what I learned, and I want to give you this. When we have load surface, like minerals in soil, say it like this: when we add more organic matter into the soil, and we form more organomineral association, the surface is gonna be more hydrophobic and less wettable. And in terms of absorption and transformation for glyphosate, we're gonna have less retention and also slower absorption and slower desorption. All of this means more transport into the groundwater or as a or runoff into the streams. For transformation, when you have higher loading at the surface, again, less transformation because absorption is the first step for transformation. When we have, so this is a kind of crucial step. Less absorption, less transformation. Also, we found the transformation became less slower. And when we have transformation, AMPA pronounced more this time compared to the surface without any organic matter. And that's not good. And yeah, that's my conclusion somehow. It's going to be problematic, high organic matter in the soil environment this way in terms of contaminant transport and contaminant fate. But the problem is the contaminant first, but this is a subsequent kind of interactions. Another bonus thing I want to share is about organic-organic interaction order between glyphosate and those high molecular weight and low molecular weight DOMs. So based on NMR studies, so high molecular weight protein is much stronger to make a non-covalent or hydrogen bonding to glyphosate compared to peroxide. And also there is no interaction between low molecular weight molecule like peptide and glyphosate. This already suggests that we need to pay particular attention to the type of DOM in evaluating the behavior of contaminants at their environmental interfaces. It could be water, it could be soil system. Here I want to acknowledge. Thank you, Anita. Past 8 years, yeah. You supported me and you led me to learn more about soil science and also to developing skills, any skills. You led me to try anything. And also my lab mates, my previous lab mates, Michael, Taylor, Ariel, and my current lab mates, Amy, Luna, and Alyssa. They are awesome. I know this is a teamwork, and it's not possible to do something like this lonely and also not supporting environments. My special committee, they had good advice, and also I minored environmental processes and environmental assessment in environmental engineering school. And I had time to learn about those things with Damien and Matthew. And I want to thank Yi and Hugh for helping me with measuring TOC, manganese, phosphate, glyphosate, not glyphosate, sorry, orthophosphate, because they have IC, by chromatography. I use CCMR facilities and different facilities at Cornell, LCMS at BTI, and measuring a surface area, zeta size, zeta potential in Energy Institute. And I've worked with Robert Page to make that microfluidic reactor. So some of those researchers and also some of those scientists, they are really supporting and inspiring for me. Linda Nicholson for supporting me to do NMR studies, and Mary McBride is can say his research and his attitude is inspiring. What I'm doing here, he started, I don't know, maybe 30 years ago to study this. I'm just adding a little piece to what he found in the past, like 30 years. Also, the people from different places that they helped me to do modeling for some stuff that are not shown here. That's the thing, like Eugene Elton helped me to do peak modeling for manganese fractions. And also my friend, Nasrullah Sebernia, helped me to do 1D transport in hydrous. And at the end, my family, my lovely wife, Riley, and adorable kids, Aiden and Lena. They made a positive environment and safe environment for me to do this research. And also my friends, Saeed, Teddy, Sonam, I think all of you know Sonam. Richard is my business friend. He's my founder. I'm running a startup company. He's my founder and his co-founder. And Michael Schmidt twice. Yeah, thank you so much. It's over, but if you have any question, I'm gonna take those questions.
Questions?
Yes.
I have a question. So the polysaccharide was diminishing the ability of the glyphosate to absorb, and the idea there is that it's going to then perhaps reduce the chemical transformation by the mineral, right?
Polysaccharide, the second act is about absorption on iron oxide. So on iron oxide, there is no transformation for glyphosate. So what we see is a pure impact on absorption only. So it diminishes absorption of glyphosate. But so I needed to work with another molecule. Actually, it's a fair question. I picked protein because I had a batch study before starting the transformation study. I picked different organics, so lignin, polysaccharide, polygalactic acid, protein, and other stuff that people can detect in soil. Mixed them with manganese oxide and started to see the transformation of organics itself before adding glyphosate. I found protein is more stable at the surface. That means less problematic for me if I want to detect the byproducts, or I'm going to have more stable surface to see a transformation for glyphosate. So that was the reason I changed from polysaccharide to protein on manganese oxides.
I guess my follow-up question there is in terms of what the behavior is in soil. It seems like to get chemical transformation, you need to get interaction with the surface.
Yeah.
But in terms of like enzymatic or microbial degradation, that's going to be maximized in solution. And so potentially you have one form of transformation is when you're on the surface, but then processes that are inhibiting that form of transformation might actually enhance microbial transformation.
Nice.
So in terms of the behavior of the system, what would be best for degrading glyphosate?
Yeah.
Do you want it to stick to the surface, or do we want it to not stick to the surface?
Yeah, that's a fair and valid fact. So it's now at the surface, more available for bacteria, and also more available for enzymes can be degraded. But those biotically instead of abiotically. Thank you. Yeah, it's good to follow up. Like now, system needs to have a microbe. How it's gonna be organomineral association and microbe. That could be something to interesting.
All right, so for the interest of time, I think thank you very much, Behrooz.
This has been a production of Cornell University, on the web at cornell.edu.